City Halid. I'm the one first reporter in which I report about the spawning in which we have three types of place in propagation in which flow, confined water, flowing water, and freshly inundated area. So we have second topic. It's about egg development in which we have 7 stages. The third one is the fate of developing eggs, and the fourth one is the course of natural spawning. So I'm the first reporter. I'm the one to talk about the spawning. So let's talk about what is spawning mean. The spawning is expulsion of gametes from body into surrounding water. This is what we call spawning. So nangyayari sa mga fishes. So we have a different type of spawning. Place of propagation. We have three types of place propagation. Spawning of the fish. This is the confined water, flowing water, and the third one is the spawning in a freshly inundated areas. So we will tackle first the confined water. This is the chosen places for spawning are often different species. So some of the species scatter the eggs on a submerged with stone or gravers. So may mga fish ni talaga na nag-scatter ng mga egg nila sa mga stone and weeds. So some of them scatter its flowing eggs in the water columns were developed further. So may mga fish din talaga na naglelay ng eggs, nag-scatter ng egg sa water column. Later on we will see some picture. Some of them lay their eggs in one place adhered together in a damp or some sort of a moss. So they naglelay sila ng eggs in a damp. May mga fish din talaga naglelay ng egg sa mga damp. So may mga species na talaga spawn in a hole in a gap, spawn in a clay in a rock. So sa mga butas ganyan po sa mga nakikita natin sa mga putikan at sa mga batuhan. So meron din tawag yung tinatawag na nest spawner. This nest spawner search and find object just like bushy roots of water, plants, or trees, rock, or etc. which they clean free for silt and debris and make their nest. So pwede sila magkaroon doon ng mga nest. Yung nest na yon galing yun some build their nest by collecting nest piece by pieces. So yung nest kino-collect nila yon by pieces. So may mga tinatawag din talaga na mouth breeding is a very effective method on parental care. So akala natin kinakain nila yung anak nila pero hindi pala yon. So there's a spawner which abandon the eggs and leaves them in a mercy nature. So may mga spawner din talaga in which nag-abandon sila ng eggs. Tapos iiwan nila pero sa safe na lugar. The second place of propagation is a flowing water. This is a flowing water has definitely advantage in that turbid, continuous moving water flooded condition very effectively protect the eggs and larvae. So the advantage of the flowing water is that turbid. So there's types na non-adhesive, floating, semi-floating, or rolling. Eggs get enough oxygen for the development and nurturing are well concealed from predators. So nagkuha sila ng oxygen for their development. The water current causes the eggs and larvae to dip downstream and toward the shore. So the water current is the one reason kung bakit yung mga eggs ng mga fishes is pumunta sa mga bottom. The third place of propagation is spawning in a freshly inundated areas. This is the freshly inundated areas place of juveniles, and for the growth of the juveniles is nearly freely for their enemies. So the water is actually warm and rich in oxygen, factor that favors the rapid development of eggs and larvae. So yung mga water nila are mainit and rich of oxygen factors. Because tapos that favor the rapid development kung saan madali silang ma-develop yung eggs and also the larvae. This is a rich of macrofauna and flora development on decaying remnants of terrestrial plants, and this provided the prey and fingerlings with abundance of foods. So this is the different type of spawning. I know it's blurred. Sorry for may module naman tayo siguro. We just need to catch up na lang. So may mga different type of spawning in which may mga bottom nest, water column, in a hole of eggs development. So the stage 1 is the primitive egg cells. This is what we call obogonium or archobogonium, are very small, their size being hardly bigger than that of other cells. 8 to 12 microns. So, namakalit, diba? They're multiplied by normal mitosis. So, na-multiply sila by normal mitosis. This is the stage 1 in which the small their size, 8 to 12 microns lang to siya. So, the stage 2, the cells grow to the size of 12 to 20 microns and the follicles begin forming around each egg cells. So, the stage 2, kung saan nag-increase na yung laki nila to 12 to 20 microns. So, the follicles begin to forming around each egg cells. No, nagkakaroon na sila ng follicles. The follicles whose function is to nurture and protect the development egg eventually become a double layers of cells. Na, yon ang follicles, yung function in which become a double layers of cells. Stage 3, during this stage, the eggs growing significantly larger to attain a size of 40 to 200 microns to become enclosed by follicles. So, kung saan magkasing laki na sila at saka ng follicles, 40 to 20 microns na siya, in which nag-attain siya ng size ng 40 to 20 micron, kung saan nagiging magkasing laki na sila with the follicles. So, the stage 4, during this stage, the production and accumulation of yolk begin. This is a process known as the vitellogenesis. So, the eggs continues to grow size of 200 to 350 microns, which is the accumulation of duds of lipoid materials in encyclome. So, this is stages the production of accumulation of the yolks begin. Kung saan yung accumulation of yolks nangyayari na siya. This is known as a vitellogenesis. Kung saan nagkakaroon ng tinatawag natin vitellogenesis. Yung the egg continues to grow as usual in size 200 to 350 micron. So the stage 5, this marks the second phases of vitellogenesis. The cytoplasm is now fully of lipoid drops and yolk production begin. The egg size reach 350 to 500. So as usual, nag-increase din talaga yung egg sa stage 5 to 350 to 500, which is cytoplasm is now full of lipid drops and yolk productions begin. So nagkakaroon na tayo ng is now yung full of lipid drops na yung cytoplasm and the yolk production is begin, in which the production of yolk kung saan nangyayari na siya. Stage 6. This is the third phases of vitellogenesis, using which is yolk phase push the diploid drops toward the edge of the cells, where two rings begin forming the nuclei. So, this is the stage 6 is at the eggs is attain size of 600 to 900 micron. So, this is what happened in their stages. Stage 7, this is a process of vitellogenesis is completed during the stage and eggs attain a size of 900 to 1000. When the yolk accumulation ends, the nuclei withdraw into the center of nucleus. A micropyle means a small opening on the egg cells developing during this stage. So, nagkakaroon the stage 7 where the process is complete na sa vitellogenesis is complete during this stage. Tapos, nag-attain ng size yung egg 900 to 1 solution in which the pioneer referring is regulated by gonadotrophin, which is formed and stored of pituitary glands or hypothesis, just as this is a two example of the pituitary glands follicle stimulating hormones, which PS, FHS, and LH luteinizing hormones. This is the continuously produced and secreted into bloodstream. On the other hand, the steroids of hormones, estrogens, secreted by the capsule of the follicles inform that brain about the stages to egg develop. So, yung follicles, nag-inform siya sa pupunta brain in which yung egg pwede namang ma-develop. When the environment changes for the better, the fish start getting to the to the sense organs or sense organs. All the necessary information about environment condition, like for example, temperature, suitable place of spawning, water current, blood presence of fish, of the positive sex, or something and so far. This sensory information accumulated in the hypothalamus give an order to our hormones, gonadotropin releasing plus hormones, GRH, to hypophysis. To release the gonadotropin into the bloodstream. The gonadotropin, this release reach the gonads and trigger the pre-ovulation process and a final ovulation. So dito na nagtitrigger yung pre-ovulation process kung saan nangyari na yung final ovulation. So the last one is the course of natural spawning. This is the first effect of the gonadotropin on the eggs that movement on its nucleus toward the microphil. This is followed by the terms of hydration where the eggs absorb water. All these are completed during the pre-ovulation stage. So, nagkakaroon sila ng absorption of water kung saan nako-complete dito yung pre-ovulation stages. This is the effect of the gonadotropin on the eggs, the movement of nucleus toward the microphil. So, nag-move yung mga nucleus toward the microphil. So, the first stage of meiosis occurs. This is where the first meiosis occurs, during which the total number of chromosomes is reduced to half. At the same time, the follicles keeping the eggs fixed on the wall of the ovary get dissolved by enzyme. So the refertilization in which eggs falls into cavity of the ovary. So the second division of meiosis normally takes place in the presence of the sperm, which intrude into the nucleus of the cell through the microphil. So, which microphil, therefore, the presence of the male prenucleus necessary for the development of female prenucleus. So, may correlation pa rin ang mga male and female prenucleus. That's all. So, the second reporter will be Take to Chime.
Good morning, ma'am. Good morning, classmate. Today, I will discuss the artificial fertilization. So, what is artificial fertilization? Artificial propagation, the females are given one or more injection of chemicals which regulate the final repairing for dormant eggs in the ovaries. As soon as the eggs are ripped, they are stripped from the females. The male are usually also inject eggs are artificially fertilized with sperm obtained from the males and reared under the controlled conduction. So, as you can see, this is the sample of the regulatory repairing of eggs. So, first is ripped eggs are stripped from the female's fish, then follow the eggs or artificial fertilized with sperm from the male fish. So, this is the last. Eggs are then reared under the controlled conductions. Parang box siya na mainit. And the natural propagation, significance of propagation. So fishes are, as other living organisms, must adapt to the environment if they are to survive and flourish. The most vital point in this struggle for existence is their ability to successfully propagate rapidly during their lifespan and add a large number of offspring to the population. The survival of, sufficient number in the face of innumerable hostile environmental factors hold the key to their success, since at this stage they are weakest and most vulnerable. So two, uh, biology of propagation. The propagation habit is the most vital adaptation of the fish to its environment with respect to survival. For successful propagation, the place where eggs are released should have optimal conduction with respect to eggs, oxygen, temperature, food, etc. should be almost free of aims. Those species unable to feed such conductions have gradually been limited. So number three is age of sexual maturity. So some fishes such as tilapia become successfully mature within a few months, while others may take as long as a few years. Sexual maturity depends on several factors. So sexual maturity is delayed in cold climate, while it accelerated in warm environment. So these are artificial propagation finfish. So a need for artificial propagation. So some kind of intervention by man in the course of natural propagation of cultivated or cultivable fishes may help to achieve a better survival of their offspring. So the techniques of artificial propagation of fish are manifold, all of which are aimed to produce plenty of spawn, prey, and fingerlings for utilization in culture or for restocking water bodies or water courses. The demand for quality fish seeds is particularly great for more than the intensive and super intensive culture system. So B, the different practices of artificial propagation. So artificial propagation as a practice in different parts of the world may vary depending on local conduction and facilities. So it may start with the collection and further rearing of natural produced egg, spawn, or fray, or which productions of egg itself with artificial inducement, followed by controlled fertilization, hatching, rearing of larvae, and fray. So as you can see, the sample picture give you a fertilized egg releasing from the artificial fertilization or hatching and relap up to the fingerling stage under controlled conduction. So C, collection and rearing of fertilized eggs, larvae, and prey. The collection of floating eggs, larvae, and prey for fishes spawning in rivers and inundated area. So in as age old practice in India and China, a large number of funnel-shaped nets are operated along gently sloping banks of river in the paths of the different difting eggs or larvae. So the producement for fertilized eggs and their rearing. So it is possible to produce fertilized eggs completely or almost free of the eggs of other fishes, such as eggs are incubated under controlled conduction, and the resultant larvae can be reared to fingerling stage. So according to the requirements of the species concerned, The goal is to achieve the best possible survival. So rate goal growth and health. The fertilized eggs can be produced in the several different ways. So last, procurement of fertilized eggs without hormone treatment. So fertilized eggs can be produced without restoring to the hormone treatment by the following method. So the collection of the eggs nets spawning by place artificial nets in their natural spawning place, imitating the natural spawning conduction in a smaller artificial pond by proving nets or grass mats to serve as egg collectors, stock board fishes than spawn. That's all. Thank you. Good morning, ma'am. Good morning, classmate. So, the next that I will discuss is procurement for fertilized eggs by inducing breeding through hormone treatment. So, producing of fertilized eggs by inducing breeding through hormone treatment. This also can be achieved by either of the following techniques. So, inducing spawning in small tanks by administering a human gonadotropin hormone. The above-mentioned techniques are simple and inexpensive without requiring any sophisticated installation or tools and without the difficulties and risk involved in the artificial fertilization. As a result, these techniques are widely used in the artificial propagation. So as you can see, this is the sample of the course of induced ovulation. So artificial fertilization and subsequent rearing. So artificial fertilization and subsequent rearing. So in artificial fertilization, the culturists handle the bored fish and it's therefore in a position to eliminate unsuitable fish and to choose the right type of fish for eventual stock improvement. Further, these techniques also enable the culturists to produce useful hybrid, combining the desirable qualities of different strain of fish of the same species of different fishes. So this is the step for artificial propagation. So step in artificial propagation, the artificial propagation of fin fishes is a chain of activities which between the various species have some point of similarity while differing in others. The various activities involved in the process of artificial propagation of fin fishes are listed below. So captured of wild brood fish from spawning grounds, selection of breeders from wild stock or natural spawning or for hormone treatment, rearing of board fishes, inducement of natural spawning with or without hormone treatment, so procurement of sexual productions by stripping with or without hormone treatment, artificial fertilization, incubation and hatching of eggs, and rearing of larvae, fry, and fingerling. So, as also indicated, not all the listed activities must be followed in the propagation of all fishes. It depends on the stage at which artificial interventions starts and the specific requirements of individual species. So the technology that is to be applied for the propagation of a given fishes depends on the spawning habits of the fish, the given of possible local conduction, the available installation equipment and tools, and the adequacy and skill of the personnel available for propagation work. Survive of artificial propagation as practiced in the different parts of the world show us even for the same species there is no hard and fast technology for artificial propagation. In most cases, The fish culturist adopts the technology which his feeds is easiest to apply and which provides successfully results. So, however, the degree of success may not be the same different methods. So that's all. Thank you.